stand between you and delicious Austin lunch, so I appreciate it. So as some of the speakers have already talked about, ecotypes are very important stages in the process of speciation. They occur in all different types of organisms, and they represent early stages in that process. What a lot of people don't know is where the term ecotype comes from. And that's from this guy named Go Turesson, who was a Swede, who back in the 1922 published a couple papers where he took plants from all over Sweden and he planted them into common gardens and found that they looked very different. So their differences were due to genetics. In particular, he was interested in planting coastal populations and inland populations in common gardens, and he always found that the coastal ones were prostrate, they grew out to the side, while the inland ones grew upright. And this was consistent across a number of different plant species. And so he came up with the term ecotype, and he advocated that this was a stage in the process of speciation. So Klausentek and Heising followed up on his work and took coastal and inland populations in California and took it to the next level. They started doing reciprocal transplant experiments where they took coastal ones, put them inland ones, put them at the coast. And from this, they could figure out that they're locally adapted to their population or to their individual habitats. And so these ecotypes occur in these different zones in nature, and they're kept in those different geographic regions because of local adaptation. And so they're reproductively isolated because of allopatry, but the allopatry is due to ecological differences. And so once you're in allopatry, then you can build up all kinds of other reproductive isolated barriers. So Klausentek and Heising also found these coastal plants that are prostrate and these inland ones that were upright. And if you make an F1 hybrid, you get sort of an intermediate one. And if you go to the F2 generation, you get ones that are upright, ones that are prostrate. And we see this pattern all around the world, but we don't know what causes it at all. We have no idea what the underlying genetic mechanisms are. So how do we get at the genetic basis of this plant ecotype formation? There's a lot of different phenotypes, including this prostrate structure, but there's all kinds of other adaptations, and each one of these probably has a complex genetic basis. So I think what we want to do is use genomics. Now the system that I'm going to talk to you about today is the yellow monkey flower, my favorite system. So coastal perennial plants, these are very large. They live out in coastal California, Oregon, and Washington. They're huge, but they have a prostrate growth. They grow stolons, and they grow roots off those stolons, whereas inland ones are annual, and they grow just pretty much upright, and they grow really quickly, and they flower before the onset of the summer drought. So they're trying to escape from that summer drought, and these differences are highly genetic. So what's really driving this, as I mentioned, is the summer drought. During the summertime in California, the coast is covered with fog, and this decreases transpiration, it lowers the temperature, and it adds soil moisture to the soil. So the plants out there can maintain this perennial life history, whereas the plants inland have an annual life history. This is what the temperatures look like in California during July. Temperatures are much colder at the coast than in the inland regions. So in order to understand this, when I started grad school, my third rotation project was to drive 18,000 miles and collect 100 populations of Mimulus mutatus all across California and Oregon. And then I was able to ask the question, do all the coastal ones collectively comprise one ecotype versus all of the inland ones? And so back in the day, all we had was microsatellite loci. And so with 10 microsatellite loci, I was pretty much able to separate out the two groups. So it looks like these are two large groups that occur across 1,300 kilometers, but they're only separated by 3 to 5 kilometers from each other. So people have come along since then and taken 38,000 SNPs. They found pretty much the same pattern as this 10 microsatellite loci. So we've confirmed that the coastal ecotype is different from these inland annual forms. So if you do reciprocal transplant experiments to test whether they are locally adapted and ecogeographically isolated, we've done reciprocal transplant experiments in those 3 different locations. And in fact, across reciprocal transplant experiments, the inland ones always survive better than the coastal ones in inland habitat, and the coastal ones always survive better in coastal habitat. And in inland habitat, escape from summer drought is a big deal. So the inland ones, 40% of them survive to flower, but out of 344 coastal plants planted, only 1 plant survived to flower, and it only produced 1 flower. So this is really strong selection. So getting back to this question, how do we get at the genetic basis of this? So what I've done done recently is I did a pooled sequencing. And this isn't your typical population pooled sequencing. I call this whole ecotype pooled sequencing. So what I did was took 50, or was 45 coastal populations, ground them all up together, and then took 52 inland populations, ground them all up together, threw them on the Illumina sequencer, got back 29 million SNPs. So that's 10,000 SNPs per dollar, which is like 23andMe, right? And so I'm going to tell you about those results, telling you about the results having to do with chromosomal inversions, and then also about this developmental evolution difference of ecotypes. So the first thing that comes out from this pooled sequencing data is that there's a lot of outliers, but there are these two main regions where we see enrichment of outliers. And these outliers turn out to be chromosomal inversions. Now one of these I've already known about for a long time, and I've shown that this inversion is fixed between coastal perennial populations, inland annual populations, and I've done reciprocal transplant experiments to actually show that this contributes to both adaptation and ecogeographic isolation in the field. So it's the only inversion that we know for sure contributes to adaptation because we've done an experiment. So if you look at this region, which is this one down here, it has highly elevated divergence. And then there's this other chromosomal inversion that was discovered recently on chromosome 5. And that region, which is this whole part of the pseudomolecule assembly, is totally screwed up because it's based off a cross that had the inversion in it. So that's why these things are all out of order. But all the scaffolds that I highlight there are elevated for divergence for this other inversion region. And so if you plot this out and look at the G statistic, this is the genome-wide. Here's the statistic for the chromosome 5 inversion and for the chromosome 8 inversion. So to me, this is great because this confirms that the method of just grinding up these two ecotype pools actually is working. So, to tell you about developmental evolution, we have these two QTLs that we knew of before, DIV1, which is one of the chromosomal inversions, which we know is adaptive, and DIV2, which is another locus on chromosome 8. So, Janice Friedman's done some really neat work with some near isogenic lines that I made, where she showed that these two loci interact epistatically. So they may be in like the same type of molecular pathway. So when I went down and looked into the inversion to look for outliers within the inversion, I actually found this gene, GA20 oxidase. And this is like a key green revolution gene that's involved in the biosynthesis of gibberellic acid. And for those of you who know plants, gibberellic acid is like the growth hormone of plants. And so this is like